last, but the last couple of questions, the last set of questions are related to the next couple of lectures. I've also posted on the Carmen site a document which will tell you when I'm having office hours next week. I've got a couple of in-person and one virtual time. If that doesn't work, and you still have questions, we can set up an appointment to meet either in person or virtually. Or don't forget about our TAs are also available. So should need lots of help in the next week. So let's shift. No more motor system, no more sensory system. We're going to, on our last three lectures, get back up into the forebrain. Today we're going to look at the hypothalamus, one structure of the diencephalon we haven't talked about. And then we'll also talk about the limbic system on Thursday and finish up with consciousness and sleep. Something we all like. Okay, again, objectives. So just a reminder, I'm sure you all remember, here's our mid-sagittal cut through the brain, pons midbrain diencephalon. And the structure we're going to talk about today is this area that is just ventral, just inferior below the thalamus proper. There actually is a little indentation there. And this is the hypothalamus. And as it says there, this is kind of a point where autonomic, endocrine, limbic information comes in. The hypothalamus can then use all of that and come up with some appropriate behavioral response. We can't possibly talk about everything the hypothalamus does, so we'll talk about a few things. It's kind of as representative. The large part of this lecture today, again, is a review of material that we've already talked about. So, last chance to get it all together, like the autonomic nervous system, and we will revisit the pineal gland briefly. So, we can define it as this area. Our key landmarks are going to be the optic chiasm, rostrally, the mammillary bodies, posteriorly, and we'll identify a few other structures. So it's kind of this whole, looks very smooth area, as I say, just ventral to the thalamus. I've already said this, it integrates somatic and visceral information, and it's a very important link between the nervous system and the endocrine system, and we will focus on that today. That's a pretty important function. Usually when we talk about the home of the hypothalamus, the term homeostasis comes to mind. And that's really what it's involved in. It's trying to make sure that we're maintaining our internal environment in a very narrow physiological range. So among all of its functions, it regulates heart rate and blood pressure. We'll touch on blood pressure a little bit. Body temperature, electrolyte balance, eating, feeding behavior, and along with feeding behavior, part of the endocrine system, our secretion of enzymes from the GI tract, and sleep cycles. And that'll be our last lecture. So we will revisit the hypothalamus on the last lecture. I'm not going to talk about sleep today. So you can see that if you had a tumor, if you had something that damaged this area, you would be really disrupting our normal homeostatic mechanisms. And that could be fatal. So tumors in this area can be very, they are usually very aggressively treated if possible. Okay. So, if it's going to play all these roles, where is it going to get input from? So here's the good news. I'm not going to tell you the names of all of the tracks that carry information, but I do want you to know the general areas that information comes from. So on this slide, you can see it gets input from the limbic system, in particular the hippocampus via the fornix. We'll talk about that on Thursday. It also gets input from the amygdala via this green pathway. Again, I'm not going to put all the names in. There's a lot of information that comes from the cerebral cortex and from that ventral part of the striatum. That ventral striatum or accumbens sends a lot of information into the hypothalamus. We already talked about input from the retina, going to the suprachiasmatic nucleus to regulate control of melatonin release from the pineal gland. And there's also, and I will show you this shortly, input that comes from the brainstem. And in particular, one of the cranial nerve nuclei that we've already talked about. And we will revisit that one shortly. And in terms of outputs, an important output is to the pituitary gland, which we'll be looking at today. Again, that is one of the things that I want to focus on a little bit. Sends input back to basically the same places that provided input. Back to the cerebral cortex, back to the limbic system. There's a projection from the mammillary body, which is part of the hypothalamus, that goes back up to the anterior nucleus of the thalamus. And from there, the anterior nucleus of the thalamus has input to the cingulate lobe, which again is part of the limbic system. And there's feedback to the brainstem and spinal cord. So it's almost kind of, you look at this, it's almost like a mirror image. Inputs coming in, outputs go back pretty much to the same areas. So know the general areas that are coming in, especially those that we're going to be talking about today. So the term sensory is coming back. If it's going to regulate body temperature, it has to get some information about what's the temperature. There are temperature-sensitive neurons in the hypothalamus. They're going to regulate that. We'll talk a little bit, like I said, about blood pressure and osmolarity of the blood. So it is getting feedback, and a lot of that is coming in from the periphery. And you can actually get 3 different kinds of responses. One is called visceromotor. Viscero, again, referring to the organs. And so one of the roles of the hypothalamus is to adjust. Remember how I kept saying, the sympathetic and the parasympathetic nervous systems, it's not always one or the other, but they're in balance. And it's up to the hypothalamus to kind of set, do I need more sympathetic tone? Do I need more parasympathetic tone? Depending on the input that I've received from some of these areas. You could have a humoral response, and that's going to be maintained or derived from input to that pituitary gland. And you could have some kind of a motor response, that feedback to the cortex. If you're shivering, your behavior might be to go put a coat on. Today, I think it's just the opposite. I've noticed everybody coming in shorts and no coats. I haven't been out since early this morning, so I'm assuming it's very nice out there right now. Remember when we started back in January and everybody came in their big coats and gloves? Always happens. It's nice to see that transition. If you're thirsty, there's areas that are going to tell you to start drinking, eating, sexual response. So this is kind of what the hypothalamus is involved in regulating. So let's review the autonomics kind of quickly, although we want you to remember this again. It's kind of important. So, same slides I used way back in January when we started out. Remember, there are two functionally, chemically, and anatomically distinct parts of the autonomic nervous system: sympathetic and parasympathetic. You can almost forget about these, but not quite yet. And my argument is always, it's not one or the other. We don't turn sympathetics only when we're in a stress panic mode. When you get up and leave, you're going to turn up your sympathetics to get a little more blood circulating. Our basic unit, when we talk about the circuit, there's a preganglionic neuron that is always going to be located somewhere in the central nervous system. And then there's a second neuron called the postganglionic neuron, which is always in a ganglion somewhere in the periphery. So unlike the somatic motor system that we talked about, where you have one motor neuron going to your skeletal muscles, in the ANS, you have to have two neurons working together. You lose either one, and you're in trouble. And it's that postganglionic neuron that actually innervates the smooth muscle or activates the glands in the periphery. Vaguely familiar? It's coming back, right? See? It's the nice thing about learning. You learn it once, you can sort of forget. The second time through, it's like, yeah, why are we talking about this now? Okay. Parasympathetic, again, just a reminder, there is no overlap in the location of the preganglionic neurons in the sympathetic and parasympathetic nervous system. For parasympathetics, they're found in the brain stem. We went through that with all the cranial nerves, or in the sacral part of the spinal cord. Okay. In contrast, the sympathetic nervous system, the preganglionic sympathetic neurons, are in the thoracic and upper lumbar levels of the spinal cord. So those two divisions, their first order neurons, are totally spatially separated. Probably makes more sense now than the first time we went through it. Same thing for the postganglionics. Sympathetics have their own location for postganglionic neurons, primarily this sympathetic chain, or a few that go down into the abdomen, and parasympathetics, with the exception of a few that we looked at with cranial nerves, the postganglionics for the parasympathetics tend to be located within the wall or very near the wall of the organ they innervate. And again, they usually have opposite effects, or it may be that only one of the systems goes to a particular structure. Sympathetics are going to increase heart rate, parasympathetics decrease, pupil of the eye, sympathetics dilate, etc. So why are we going back over all of this again? Well, everything we've talked about, it's kind of like when we talked about the somatic motor neurons. We said they could go out and innervate the muscle as long as they got some kind of input from the proprioceptors. They could function. You know, we can get contractions of the muscles at the level of the spinal cord, very reflex. But we added on things like the motor cortex, the reticular formation, the vestibular system, which control those neurons. So, what's controlling the autonomic nervous system? These are just neurons in the spinal cord. They're not getting a lot of important information. Well, the hypothalamus is. So it is the structure, it is the upper level control that's saying, do we need more sympathetic? Do we need more parasympathetic tone? And a very important part of that system, anybody remember the funny little nucleus that we call the nucleus of the solitary tract? You looked at it on a cross-section, those two little kind of eyes looking out at you from the medulla. We're coming back to it. You may or may not remember, but the nucleus of the solitary tract is kind of this long nucleus that extends from the rostral medulla kind of up into the mid-pontine levels. And the rostral part, this part they're showing in yellow, gets all of the taste information, regardless of which cranial nerve is carrying it, 7, 9, 10. All of those axons will terminate in that rostral part. And the caudal part that we see here in green is getting input from visceral afferents, and in particular from cranial nerves 9 and 10, the glossopharyngeal and the vagus. Remember, our vagus nerve is going all the way down into the abdomen, so it's going to bring back information on distension of the gut. It's going to bring back information on what enzymes are being released. Is there pain in the gut? And we can see that those visceral afferents, there's their little ganglion cells, related to 9 and 10, bring that information into the caudal part of the nucleus solitarius. There are some second-order visceral afferents, and these are primarily for carrying pain information from the viscera. And they probably primarily they're going to come from the same neurons that gave rise to the spinothalamic tract, the dorsal horn, the neurons in the dorsal horn of the spinal cord. So your visceral pain afferents will come in, synapse on those dorsal horn neurons, and if they're coming from the viscera, they kind of jump off the spinothalamic tract and go to the nucleus solitarius. So, let's make a little diagram here, looking at this higher level control. So, again, it's kind of a reflex. Here's visceral sensory input. Comes up, synapses on neurons in the nucleus of the solitary tract. From there, information can go two ways. We can complete a reflex loop, and those solitary neurons will project back down to preganglionic neurons, sympathetic and parasympathetic, and we can elicit a visceral motor response, kind of like right here at the level of the brainstem. But the nucleus of the solitary tract is also sending that information up to the hypothalamus. Remember I showed you on that first diagram of afferents, there were brainstem inputs? This is what we were talking about. We're talking about these inputs coming in from solitarius. And then the hypothalamus, among other things, can also contribute and produce a hormonal or some kind of behavioral response through its output to a variety of areas. So here's our little cross-section. Here's our solitary nucleus sitting out here. Information coming in from the spinal cord, coming in from cranial nerves. Some of these are taste inputs. 9 and 10 would be carrying that visceral sensory information. Output going to the hypothalamus. Don't worry about this parabrachial nucleus. And then we also see output Solitary can go back to 10, which is our primary parasympathetic output, or back down here to the spinal cord. So here's a really complicated looking diagram of what's really going on. Don't start writing all of this down yet. Okay? But basically, it's what we just looked at. You know, here's our smooth muscles and glands, visceral information, solitary tract, ganglionic, going back to those preganglionic or postganglionic, and we have that loop. We can also go up to higher level centers in the brainstem to the hypothalamus. But let's simplify it a little bit for our purposes. I just want you to know that nothing is really as simple as I'm telling you in the brain. But, you know, if you go out and become a hypothalamic autonomic nervous system expert, you can say, boy, you never told us all that. You can come back and lecture to the class. But this is kind of the base circuit that we want to work with. I'm going to bring this back 1 minute. When we talk about the limbic system, we will talk about the amygdala input and what that means to the hypothalamus. And the cortex that we are going to be concerned with here is the insular cortex. Remember that hidden cortex under the frontal lobe and the parietal lobe, kind of buried deep. That's where a lot of that visceral sensory information ultimately winds up. And there's part of the prefrontal cortex where we're going to see some visceral motor output. So, what would happen if we disrupted this circuit? And there's a classic disorder where that happens. It's called Horner's syndrome. This one you should pay attention to. Okay. And let's look at the symptoms first. It's primarily an effect on the sympathetic nervous system. And it usually involves disruption of that descending input from the hypothalamus to the preganglionic neurons. There's other places we can elicit it. So here's the symptoms. Drooping of the eyelid. It's called ptosis. We didn't really talk about this very much, but in your eyelid, I mean, we talked about a muscle called the supralevator palpebrae. That's a somatic muscle. I mean, I can open my eyes and I can close them voluntarily. But we also have a small smooth muscle in our eyelid that gets sympathetic innervation. And if you lose the innervation to that little muscle, it's hard to keep your eyelid open. So it's kind of there as a support. On the side of the lesion, we would have pupillary constriction. Look at the pupil here compared to this side. Remember, the sympathetic nervous system dilates the pupil. So if you lose the innervation, now there's nothing to counteract the parasympathetic innervation, which constricts the pupil. So that balance gets messed up. You get decreased sweating on the ipsilateral side. Sweat glands are only innervated by sympathetic neurons. Remember, there's no parasympathetics going out to the periphery. Parasympathetics pretty much stay internal. Decreased vascular tone. Why would I have decreased vascular tone? What does sympathetics have to do with the tone of my blood vessels? Anybody remember?
They constrict them?
Right. They constrict your blood vessels. So if I lose my sympathetics, the blood vessels relax. Now they're wider and you're going to get flushing. The skin is going to look very red. So here's kind of a classic damage to the sympathetic nervous system set of symptoms. So let's look at this. So preganglionic sympathetics, especially in the upper thoracic cord, they're controlling, they're going to send, so here's our preganglionic neurons, these little red guys down here. Their axons leave the cord, they enter the sympathetic chain, and they come all the way up to the very top of the chain. And there's a ganglion there called the superior cervical ganglion. The postganglionics leave, and a lot of times they'll just kind of follow the blood vessels. So here's one coming up with the carotid artery going to that dilator pupillae muscle. Others will go out to the periphery to innervate our blood vessels and our sweat glands. So now, what if we have damage somewhere along this pathway? So one place you might have it is this tract. So here's the hypothalamus. Here's the descending output. Coming down, could be going to solitary nucleus, or it could be actually coming down to these preganglionic neurons. So say you have a stroke, a Pontine stroke, or you have a tumor anywhere along that pathway that disrupts that descending hypothalamic input. So now there's no descending control to those neurons. Another place, maybe you have a spinal cord injury in the thoracic spinal cord. So now I could be directly damaging both the descending pathway and the preganglionic neurons. And the third place you can get damage would be at the level of the superior cervical ganglion itself. It comes all the way up, kind of sits toward the base of your skull. How might that happen? Well, if you had some kind of damage, gunshot, stab wound to your neck, you could damage either the chain itself or that superior cervical ganglion. It's usually where you see it is when you have tumors that are right at the apex of the lung. Your lung actually pops up a little bit above your clavicle, and that can put pressure on that sympathetic chain. Tumors of the thyroid gland can grow and involve that, or any of the cervical lymph nodes. So there's kind of a classic case of what happens when you lose one half of your autonomics. So I do want you to pay attention to orders a little bit. Okay, we're done with the autonomics. And now what we want to do is shift a little bit to more of the endocrine function. So we took care of that. Now, let's talk about endocrine. Before we do that, I have this in here. We just said the hypothalamus plays a lot of roles. And just like other areas of the brain, it has many little subnuclei that are specialists. So it's not like every cell in the hypothalamus is doing everything. There are specific nuclei that take care of it. So again, we're going to do this pretty generally. So here's a sagittal view. What are our key landmarks? Key landmarks for identifying areas are going to be the optic chiasm, the mammillary bodies, and this little strip of tissue between the mammillary body and a stalk that's going to be the stalk of the pituitary gland. And that's referred to as the tuber cinereum. So here's on a real image of the brain, the optic chiasm, the mammillary body, and this tissue coming toward the infundibulum, which is called the tuber cinereum. And when we look here, what we can see is they sometimes will divide the hypothalamus into three anterior to posterior divisions. The anterior division sits right over the optic chiasm. The posterior is related to the mammillary bodies, because they're the most posterior part. And then in between, you have what's called a tuberal region, because it's related to that tuber cinereum. Okay, so that kind of makes sense. Optic chiasm, tuber cinereum, mammillary body, anterior, tuberal, posterior. And we did that, in the cerebellum. We divided it into medial to lateral, vermis, medial hemisphere, lateral hemisphere. And we also said there was an organization from rostral to caudal. And that's also true in the hypothalamus. So now let's take a transverse section, and these divisions are going to go through these sagittal zones. Again, kind of intuitive. So here's the third ventricle, and there's an area immediately adjacent called the periventricular area, and then we have a medial and a lateral zone. So it's kind of like a grid. You've got an anterior, posterior, and you have a medial, lateral, and you can have different nuclei in the grid. And people have put together these little tables telling you, if you look here, this is a lateral area, or the medial area. No, I'm sorry, lateral. They've taken it. And so here are nuclei that are anterior, tuberal, and posterior. I will not ask you to tell me what are the coordinates for a particular nucleus. But I do want you to know that there is an organization and that there are many little subnuclei that make up this area. We are going to focus on a few of them. We've already talked about that one. Suprachiasmatic is telling you where it is, so even if you had to guess, I would hope you would say it's probably in the anterior zone, because it's sitting on top of the chiasm. But I'm not going to ask you that. Okay? That's a little too picky. So we're going to review that one more time, our circadian cycle. There's another nucleus that's in that two nuclei that are in this area called the paraventricular. Now that, you have to know, it's not periventricular, it is para. Para means adjacent. Peri kind of means the same thing. So if you were asked to write something in and you put in peri and it was really para, I wouldn't be able to give you credit. So very different names. Okay? So we're going to talk about these two because these are the ones that are primarily involved in the endocrine function. So here's our paraventricular nucleus and supraoptic. Again, one is on top of the chiasm, one's right above it as supraoptic. They could give them the same names. There's another little nucleus that sits right here at the base of the third ventricle called the arcuate nucleus. And tucked over here is the lateral. And we will talk about those at the end today because those are two nuclei that are involved in feeding behavior. And then there's a couple of others that you don't really see on this particular slide. The lateral preoptic, and a tuberomammillary, which are involved in our sleep-wake cycle. And we won't talk about those until next Tuesday. So today we're going to focus, we're going to review suprachiasmatic, look at the paraventricular and the supraoptic, and the arcuate and the lateral region. Okay. Pineal gland, a little bit of review. Unpaired glandular structure. Most posterior part of the diencephalon. It's the structure, it's unpaired, and it is the structure that secretes melatonin. We did this once before, but let's go ahead and review it one more time. It is under sympathetic control. Preganglionic sympathetic neurons are in the upper part of the thoracic cord. You may have remembered a question from exam 1 or 2 that asked you where were the preganglionic neurons that regulate melatonin release? And a very popular answer on that question was suprachiasmatic nucleus. The suprachiasmatic nucleus does not contain preganglionic sympathetic neurons. It regulates those neurons, but we only have preganglionics in the thoracic spinal cord. I was a little surprised, but be that as it may. So I'm just correcting that right now. Going forward. They innervate neurons in that superior cervical ganglion, which send their axons to the pineal gland to induce release of melatonin. Okay? There's our base circuit. So, now let's throw the hypothalamus into the mix. Light comes into the eye, and we know that there is a special population. Normally we would go to the optic tract, to the thalamus, back to visual cortex. But there is a subpopulation of the retinal ganglion cells that are actually activated directly by the light. That light does not have to go through the rods and the cones, down through the bipolar cells to activate the ganglion cells. They are directly activated. And those retinal ganglion cells send their input into the suprachiasmatic nucleus. I'd have to back up too far. If you go back to the previous slide, you can see now how that suprachiasmatic nucleus is sitting right on top of the optic chiasm. So very easy for those axons to just pop out and activate those neurons. Now, it's not a direct pathway, but through a circuit within the hypothalamus, activation of the SCN ultimately leads to inhibition of the preganglionic sympathetic neurons. If we inhibit the preganglionics, there's no excitatory drive on the postganglionics. There's no excitation of the pineal glandular cells, and there's no release of melatonin. And again, there it is in word form, if you don't like my diagrams. So, any questions on that? I'm reviewing it again. So now, if we're talking about the hypothalamus, we want to kind of give it its due. that's one gland it controls. The pituitary is a much more direct control of the hypothalamus. We don't have to involve sympathetics or parasympathetics in this case. This is endocrine release. Okay? So let's start with the pituitary gland. So here's an MRI. Here's the hypothalamus. There's, look here in the diagram, there's a little stalk that extends from the hypothalamus inferiorly. It's called the infundibulum. And we get to this structure called the pituitary. And the pituitary is actually two totally different structures. If we look here, so let's look at the MRI first. Here's this gland right here. Here's the anterior part of the gland. Here's the posterior part of the pituitary. And they have very different, even on an MRI, they look different. So let's look at them diagrammatically. The posterior pituitary is actually an outgrowth of the hypothalamus itself. Important concept. Posterior pituitary is a direct outgrowth of the hypothalamus. The anterior pituitary actually has glandular tissue in it. There is no glandular tissue in the posterior pituitary. So technically, even though these two are together, it's only the anterior part that is a gland. We'll talk about that in a few minutes. And the hypothalamus is going to control release of various hormones from both parts. But it does it in two very different mechanisms. Okay, I think this is the same thing. This is just a little bit bigger drawing. But the two nuclei we're going to talk about are the paraventricular and the supraoptic nuclei. And we're going to see how they control this release. Within those two nuclei, there are two types of neurons. One group are called the magnocellular neurons, big cells, 20 to 40 microns. These cells actually produce the hormone oxytocin or the hormone vasopressin. And they release those hormones into that posterior pituitary. Again, which is actually another area of the hypothalamus. Within those two nuclei, there's another group of cells called parvicellular. They're little, 10 to 15 microns. They produce what are called releasing or inhibiting hormones that then go down into the anterior pituitary and either inhibit or activate release of specific hormones. Okay, let's look at that. What are we talking about here? So posterior, let's do posterior first. Posterior pituitary is also called the neurohypophysis because it is neural tissue. And here's our paraventricular supraoptic neurons, and these are big, nice big cells. And you can kind of think about the oxytocin and the vasopressin as being their neurotransmitter. You usually don't use that term with these. But they're just like another cell would make glutamate, or GABA, or serotonin. These cells produce these two hormones. And notice the axons of these big cells come down through that infundibulum, through the pituitary stalk, and they go directly into the posterior lobe. Again, this is just another part of the hypothalamus. And what you're going to find in this posterior area is this very dense plexus of capillaries. And notice the endings of these axons are terminating actually on that capillary bed. So when we generate, there it is up there, I thought I had it separate. If we generate an action potential will come down, and just like an action potential would cause release of glutamate or GABA or whatever neurotransmitter, in this case, it induces release of one of these hormones. And they're literally forming a synapse, quote, on those capillaries. So when they release it, it goes right into the capillary bed, and from there, it will go out to the rest of the body via the circulation. Does that make sense? Questions on that? It's kind of another diagram showing you the same thing. Paraventricular, supraoptic, here's their axons coming down through the pituitary stalk, releasing these peptides, or these hormones, rather not peptides, hormones, into the capillary bed, and then out into the general circulation. So, oxytocin. It's a Greek word meaning rapid birth, and oxytocin has been most correlated with the later stages of giving birth, causes the uterus to contract, stimulates milk production. But we also know that it has a lot of roles in bonding, social recognition, empathy, maternal behaviors. Sometimes it's referred to as the love hormone. It's released when you find that perfect match and you have that bonding experience. And it's thought that the people who don't secrete oxytocin don't have much, are not very empathetic, and may even be related to some behavior, some psychotic type narcissism, trying to manipulate people for your own purposes. So it's kind of what we need to get along in society. The other one is vasopressin. Vasopressin is sometimes called the antidiuretic hormone, or ADH. And this hormone causes reabsorption of water into the kidney. So it's going to decrease urine output, and it's involved in regulating then our blood volume and salt concentration. So again, we see a little diagram here. We have pressure receptors in our large arteries. Oh, here's our vagus nerve sending information. Oh, nucleus solitarius gets back in there again. Solitarius sends that information to the hypothalamus. The hypothalamus will determine whether we need to bring more fluid back. If you have very low blood pressure, you want to bring that fluid volume up a little bit. And we'll release the vasopressin to cause fluid to be reabsorbed. The other thing it works in is detecting osmolarity, how concentrated are especially salts in our blood. So if you're recording from a supraoptic neuron, and if you very low osmolarity, we don't see too much activity. But if you start pumping up the osmolarity, that cell begins to fire like crazy. It wants more fluid to come back into the system to dilute whatever it is that's causing that osmolarity to go up so high, usually salts. And this also involves kind of another behavioral response in that it'll go, and we'll talk about this, the lateral hypothalamus and feeding and drinking behavior, so that we feel thirsty and we begin drinking more water or whatever, beverage of choice. Okay, what about the anterior, the real gland? The anterior pituitary gland is referred to as the adenohypophysis. Hypophysis was kind of the old word for pituitary gland for a long time. We just refer to it that way. Okay, so first thing to notice, it does not have a direct continuation with the hypothalamus, like our posterior pituitary did. And within the anterior pituitary, we see all these cells, little green cells and yellow cells and blue cells and brown cells, and each one of those cells is responsible for secreting a different hormone. And it has to get activated or inhibited in some cases. So what's going to control that? And those are going to be this next group of cells, which we're going to look at, which are the parvicellular cells in those same nuclei. They're just showing some of them here. Two things to notice. First of all, around the stalk of the pituitary, there is another plexus of blood vessels. And many of those vessels, notice, continue down into the anterior pituitary, forming another plexus within the anterior pituitary itself around those glandular cells. And the glandular cells are going to use that as their route to get their hormones out to the body. So that's something the anterior and posterior have in common. They use the vascular system to get their substances out. So what's happening here? Well, first of all, here's our little purple axons, and notice they don't have a route to get down into the anterior pituitary. Again, there's no connectivity in this area. But what they do have is this capillary plexus that's right at the base of the hypothalamus. And notice the axons of these cells are forming, again, I put in quotes, synapses on these blood vessels. You generate an action potential. Oops, we can do it on this one. Take it back here and then I'll go into one. You generate an action potential. They release either what's called a releasing or an inhibiting factor. That factor uses this blood vessel highway to get it down into the anterior pituitary, and whichever glandular cells have the appropriate receptor, they will bind that factor, and they will either release or stop releasing their specific hormone. If they release it, it's going to come out into the bloodstream, and here's kind of a list. I've got another list for you of some of the hormones that get released. Again, here's kind of that other picture you can see better. Here's our little cells sitting and dumping their releasing factor into the capillaries, comes back down into the anterior pituitary, and causes the appropriate hormones to be released depending on what receptors they have. Lots of different hormones. Follicle stimulating, a lot have to do with reproduction, the thyroid glands, stress axis, growth hormones, prolactin for mammary glands. Again, we don't have time to go through all of those, so let's use just one as an example. Okay, and that's the only one I'll expect you to at least be able to go through this one circuit. This is called the hypothalamus pituitary adrenal, or HPA axis. And this is usually what happens when you're in stress. They call it the stress axis. And the idea here is that we want to ultimately release cortisol to help reduce this stress. So, here's our hypothalamus. Those little neurons, one population, release a peptide called corticotropin releasing factor, sometimes called corticotropin releasing hormone, CRF or CRH. Some kind of physical, emotional, psychological stress. And that's going to go into that first capillary bed. CRF then comes down into the pituitary gland, and it will bind to cells that release ACTH, or adrenocorticotropic hormone. ACTH gets into the bloodstream, and it's carried to the adrenal gland, which actually sits on top of your kidney. And it binds to cells, and the adrenal gland then releases cortisol. And again, that's going to be released into the bloodstream. So cortisol gets released. One of the things it does, it actually feeds back to the hypothalamus, and it's a negative feedback, meaning it blocks further release of CRF. You don't want to get cortisol building up and building up and building up. There's got to be some regulation. It'll go to other regions of the brain, and it can have an effect. I didn't tell you this, and you don't have to know this, but there is a tremendous amount of corticotropin releasing factor in the cerebellum. And that was an area that I worked on for a very long time, trying to figure out what it was doing. And it was modulating the activity of the neurons. When CRF is present, those neurons are more active. You don't have to know that, but I'm just sharing. We first of all, we identify and we give these substances names based on where they were first discovered. And then we start looking in other areas and you find out, hey, they're there too. They're not doing the same thing, but they are there. And finally, we know cortisol can go and affect our muscles. Initially, maybe in a positive way, it'll bulk them up a little bit, but ultimately, it's not very good for us to have too much cortisol. And we might be doing more damage, especially to things like heart muscles. So that's why it's good to have this negative feedback. And again, I'm just putting this in because growth hormone is another one that if it isn't regulated, if the cells are producing too much growth hormone, and if that is happening before all your bone growth plates fuse, you get this individuals who are very, very tall. If it happens after all of our growth plates have fused, The bones can't elongate anymore, but they keep building up and they get larger. They get to be huge. So you have this wrestling person here. I think is that Andre? Somebody told me that once. I'm not a wrestling fanatic. So that's what he probably has, some growth hormone instability at some point. That was just there. Okay, let's finish up with eating. One last topic to talk about. So this was really looked at way back in the 1940s. And again, the technique of choice was let's cut out part of the brain and see what happens. What are the results? So if they lesioned nuclei located laterally in the hypothalamus, animals stopped eating. They became anorexic. Conclusion, here's the feeding center. This is where hunger resides. And then if they lesion the ventromedial part of the hypothalamus, animals overate and became obese. We found the satiety center. Nothing is ever that easy. You lesion, you're cutting all kinds of things. Wasn't too far off, though. I can tell you that. A lot of studies are being done now with this agent called leptin, which is secreted by your fat cells, your adipose tissue, and it increases after eating. So leptin leads after eating. And there are leptin receptors. Remember I said arcuate, here's another nucleus. It sits right at the base of the third ventricle. And there are leptin receptors on those neurons. So here's the arcuate nucleus. And when they bind leptin, and this is a subset of, again, of these neurons, one of the things they do is they project to that lateral hypothalamic area. And they inhibit those neurons. So rather than lesioning, I'm now just shutting them down, and the animals stop feeding. They also project to that paraventricular nucleus, which is releasing things like ACTH and thyrotropin hormones, which raises the metabolic rate of cells in the body. And there's a projection down to the preganglionic sympathetic neurons, which will then, again, activate appropriate digestive activities. So now we're beginning to actually, you cut it out, you see something, now they're looking at what is the actual circuit that's involved. There's other factors that can also give you this sense of satiety and stopping eating. Again, here's our nucleus of the solitary tract getting involved. You can have stretch receptors in your GI tract that are carried by the vagus nerve to the solitary tract that gets sent up to the hypothalamus, and in particular inhibiting those neurons again in the lateral hypothalamus, saying you're full. Cholecystokinin is a peptide that gets released, and its primary thing is that it will potentiate the activity of those afferents. I'll tell you that CCK also is found in other parts of the brain, and it has nothing to do with feeding. So what if I decrease leptin levels? So now notice these cells are little diamonds, the other ones are little circles, so this is a new subset. And their role is to excite neurons in the lateral hypothalamus and to inhibit those neurons in the paraventricular nucleus so that we then initiate feeding So a lot of work now is looking at these arcuate neurons, left role of leptin. Ghrelin is another one that's secreted when your stomach is empty. Also activates those arcuate neurons that excite the lateral hypothalamus. I can always remember ghrelin happens when your stomach is growling. So you can know that it's being secreted when you're empty and hungry. Growling ghrelin. And you know, they do a lot of bypass surgery. And one of the ideas, you know, besides the fact that your stomach is smaller, is that you're also disrupting this ghrelin pathway. And therefore you don't want to eat as much. I think that's where we'll take it.